Good morning. We are again in the same chapter that is biomolecules. In the previous class, we have discussed the structure of alpha glucose and that of beta glucose. In today's class, we are going to discuss about fructose. It is represented by a six-membered ring as follows. Here we have beta D fructose and furanose structure. As you could see in beta D fructose, the carbon number 6, carbon number 5, carbon number 4, carbon number 3. Up till these 3, 4, 5 and 6, all of them have the same configuration. Okay. Carbon 2 has a ketonic group, carbon 1 has a hydroxy group. Okay. Carbon 1 have CH2OH group and carbon 1 over here has a aldehyde group. That is the basic difference in the structure of these two compounds, beta D fructose and furanose structure. Now we know that fructose is assigned a furanose structure. That is, this is the two basic structure of beta D fructose and furanose. Okay. After that we have lactose. It is made up of a molecule of galactose, and the units are represented as follows. The units are represented together, they are linked together. As you could see the structure of lactose, you see in structure of lactose what we see that we have two six-membered rings, and these six-membered rings are joined by a glycosidic linkage. What type of linkage is this? This is actually a type of a glycosidic linkage. In this glycosidic linkage, what we see, it is connected with CH2OH over here. See, both of them have the CH2OH in the same position. Here we have hydroxyl group, here also hydroxyl group, here also hydroxyl group, but this hydroxyl group is involved in the glycosidic linkage. Here we have OH, here also we have OH, and here also OH. So OH, 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 and here also OH, here OH, OH, CH. This is how the lactose, both the rings are joined. After that we have maltose. It has the structure as shown below. So that was the structure of lactose. See the structure. We have these two six-membered ring structure. Okay, and this is joined by a glycosidic linkage. After that we have maltose. It has the structure as shown below. Next we have the structure of maltose. It has the structure as shown below. On treatment with acid or with enzyme, maltose gets hydrolyzed to molecules, and these two molecules, that is alpha and beta alpha D glucose. So since one of the glucose unit exists in a hemiacetal form, it is a reducing sugar. So what type of sugar is that? That is actually type of a reducing sugar. It is a type of a reducing sugar. In that, what we see that we have these two type of structure. As you could see, two of the glucose units. Here we have CH2OH or CH2OH. Here it is joined by a glycosidic linkage. Both the hydrogen are symmetrical, and here also oxygen. Here we see that here it is OH, H, here H, OH, but here we have H, OH. This OH, H is same, and H, OH is also same over here. So that is how the structure of maltose actually looks, right? See, here one important term comes over here, that is called as reducing sugar. Now the question is, what is it reducing sugar? You should know that those carbohydrates which contains a free aldehydic or ketonic group, if it contains a free aldehydic or a ketonic group and it is able to reduce failing solution, both A and B, and Tollens reagent, they are called as type of reducing sugar. If it is able to reduce both Tollens reagent and failing solution, both type A and B, that is example of a type of reducing sugar. All monosaccharides, maltose and lactose, they are actually behaves as reducing sugars. all monosaccharide, maltose and lactose, they behave as reducing sugar. So in this case, actually what is happening, it is behaving as a reducing sugar. After that, we are going to study about sucrose. After that, we are going to study the structure of sucrose. In case of sucrose, what we know, just see, sucrose, it has a structure as shown below. Okay, this is the structure of sucrose. So in this structure, what we could see, on hydrolysis with dilute mineral acid or with enzyme invertase, sucrose gives equal amounts. Okay, sucrose and glucose are the dextrorotatory while fructose is levorotatory. Just remember, it is dextrorotatory while it is actually levorotatory. Thus, in the process is accompanied by the inversion of optically active. So the process of inversion optically active takes place. That is we could just see that in this case now what is happening, the mixture formed is invert sugar, that is sucrose, glucose plus fructose. It contains sucrose, glucose plus fructose. As you could see the structure of sucrose, see the structure. In case of sucrose, what happens, the structure looks like this. In this structure, what we see, that here we have the glucose unit, here we have the fructose unit. Okay, we have the glucose unit as well as the fructose unit, and they are joined by this glycosidic linkage. Okay, they are joined by this glycosidic linkage. In case of glucose, you can say 6-membered ring. In case of fructose, it is a 5-membered ring. And here in this position, we have a CH2OH group. Here also we have a CH2OH group. Here both the OHs are in this pattern. Here OH is alternate to it, and here also this OH is alternate, and this is having a CH2OH group. So this is actually the structure of a sucrose. Now I had told you that there are two types of sugars are there. One is called reducing sugar. As I told you, these are the sugars which actually reduces filling solution as well as tolerance reagent. But there are many type of sugars which do not have free aldehydic and ketonic group, and as a result they do not just reduce filling solution and tolerance reagent. They are called as non-reducing sugar. They are called as non-reducing sugar. Okay. After that, we will discuss the next topic, that is the sweetness of sugar. We know in general that sugars are sweet in nature. Now the question comes, why this type of sugars are sweet? What makes the sweetness in sugar? All the monosaccharides and disaccharides are generally, they are sweet in taste. Hence they are actually sweet in taste. Hence they are known as you could see, they are known as actually sugars. Okay, all the monosaccharides and disaccharides are sweet in taste, hence they are generally called as sugars. Sucrose is given the sweetness value of 100. The sweetness of other sugar is compared with the value of sucrose. The sweetness of other sugars is compared with the value of sucrose. The sweetness of fructose is 173, invert sugar 130, sucrose 100, glucose 74, galactose 32, maltose 32, and that of lactose is 16. This is the sweetness of fructose. After that, all the monosaccharides and disaccharides are reducing agents due to hemiacetal and hemiketal forms, which easily change it to aldehydic form in the alkaline medium. We change into the aldehydic form in the alkaline medium. Okay, so what we know, that all the monosaccharides and disaccharides are reducing agent due to the hemiacetal and hemiketal forms, which easily change it to aldehydic form in the alkaline medium. So just remember, in the alkaline medium they actually shows this type of property. Although fructose does not contain any aldehydic group, yet it gives tolerance reagent test and failing solution test, because under the basic condition of the reagent the fructose gets converted into a mixture of glucose and mannose, both of which contain aldehyde group. So just remember, both of them contain aldehyde group, and this is called as Lowry de Bruin van Inkensten rearrangement. As you could see that we have the D fructose and the structure is like this: OH, both carbon 6 and carbon 6, carbon 5, carbon 4 is having this OH groups over here. Okay, now when you are reacting with the base, it changes to glucose. So what is in case of D glucose? We get aldehyde group. Okay, and in case of D mannose, what we get this type of ketonic group. Okay, in this way, now what is happening, that is how the reaction proceeds. Okay, but when the same thing is made to react with the base, it changes to D mannose. So see the structure of D mannose and that of glucose, only the change in the position of the OH group, the carbon present in the second carbon that contains the OH group, that two changes are taking place. This is how it actually proceeds. Okay, this is how it actually proceeds. That is the structure of D glucose and D fructose. See the structure, same type of here also aldehyde group, here also aldehyde group, only change is OH, and both of them takes place in a basic medium. Alpha and beta glucose reacts with one molecule of ethanol to form the corresponding methyl glycoside. Okay, in this way the reaction actually proceeds. Now another thing we know, that when this type of glucose is treated with methanol in the presence of HCl, the hemiacetal form changes to the acetal form. The hemiacetal form changes into the acetal form when it is made to react in presence of ethanol in presence of HCl. Now starch, it serves as a storage for polysaccharides in plants. It serves as a storage for the polysaccharide in plants. It consists of two components of alpha glucose. Amylase. After that we will discuss about amylase. It is a linear polymer of glucose and is soluble in water. So first of all, we should know that it is a linear polymer of glucose and is soluble in water. Its percentage in starch is about 10 to 20%. Its percentage in case of starch is actually 10 to 20%. See the value ranges from 10 to 20. These are linked together by alpha linkage involved carbon 1 of glucose unit to carbon 4 of the other. As you could see over here, in this case, just see the structure. The structure we see is here. This is the structure of the normal glucose. Here it is alpha 1,4 glycosidic linkage. Why? Because the first carbon and the fourth carbon of the other is getting linked. In this way we get a type of long chain. This long chain is the structure of alpha 1,4 glycosidic bonds. Then we will going to discuss amylopectin. What is amylopectin? Amylopectin is the branch chain polymer of alpha glucose and is insoluble in water. See, the previous one was soluble, but this one is insoluble. Okay, there are about 1000 units of glucose. If 1000 units of glucose is joined one after the other to form a huge polymeric chain, that polymeric chain is called as amylopectin. See, it is a branch chain polymer. As you could see here, it is alpha 1,6 glycosidic bond. Why? Because first carbon is joined with the sixth carbon of the other chain. In this way we have a type of polymer and here we have both the 1,6 glycosidic bond as well as 1,4. See, 1,4 is in between the carbon, in between the rings of the same chain, and 1,6 in between the different rings of the different chain. Different rings of different chain, we have 1,6 glycosidic bond, here we have 1,4 glycosidic bond. In this way the structure of amylopectin has been found. Okay, so that is how the structure of cellulose has been shown. Okay, that is how the structure of cellulose has been shown. Now we are going to study the structure of cellulose. We are going to study the chemistry of cellulose. Now cellulose, as you could see, is found in all plants. It is found in all plants. It consists of 50% total organic matter of the living beings. See, it is actually made up of 50% total organic matter in the living world. Okay, in this way we can say that we have this type of polymer. 50% total organic matter in the living world is made up of cellulose. Cotton is pure form of cellulose. Cotton is pure form of cellulose. Cellulose is a linear polymer of beta D glucose. Cellulose is a linear polymer of beta D glucose. Okay, cellulose is a linear polymer of beta D glucose. This is how the structure looks. After that, you see the next one we have, that is the chains are arranged in this way. Now when it is treated, now what we know, that the chains are arranged to form the bundles and are linked together by hydrogen bond between the glucose molecules or adjacent organic solvents. Now when it is being treated with concentrated sulfuric acid in cold, it slowly passes into solution. This solution, when diluted with water, gives starch-like substances, amyloid, which is known as parchment paper. On boiling with water, it is hydrolyzed into D-glucose. On boiling with water, it is hydrolyzed into D-glucose. Another characteristic property is that cellulose gives many useful products when treated with different chemicals like rayon, gum, and cotton. Now, cellulose is directly used in making cloth and paper. Another important use of cellulose is it is used directly to make, that is called cloth and paper. Here we have the structure of cellulose. Okay, see the structure of cellulose. See here also the glycosidic linkage. So we have the same type of glucose units. They are joined one after the other, and this huge long polymeric chain is formed, and the whole chain goes on continuing. That is the polymeric structure of glucose. After that, next one we have is glycogen. Now what is glycogen? Glycogen is actually, in glycogen there are 25 glucose units, and the structure is assigned as a condensation polymer of alpha glucose, and it is considered as a condensation polymer of alpha glucose. Now, glycogen in short term food storage in animals. So in case of animals, the food is stored in the form of glycogen. Okay, glycogen, the food is stored in animals. After that, we are going to discuss the test of carbon. What are the chemical tests? For the test, we have the Molish test is performed. In the Molish test, what we do, the Molish reagent is used, in which a 10% alcoholic solution alpha naphthol and the solution of the carbohydrate is being heated, and followed by concentrated sulfuric acid along the sides of the tube. As a result, a violet colored ring is formed. This violet colored ring actually confirms the junction of these two layers. Violet colored ring, they confirm the junction of these two liquids. Okay, these two layers. As a result, a violet ring is formed at the junction of the two layers. Violet colored ring is from junction of the two layers. This is how the structure of carbohydrates takes place. Okay. Okay, children, that was all for the day. We will discuss the next part of the chapter, that is protein, in our next class. Till then, children, goodbye.